Thank you, Greg, and to the organizers for this wonderful invitation to talk about our work. As Greg mentioned, I'm a microbiologist by training, and I'm learning as I go in terms of autism. And what I'll talk to you about today is very much an early stage research program. And again, Dr. Gordon mentioned the importance of research. We work with mice. And so oftentimes I'll wear a name badge that says Sarkis Mazmanian, MD. I often joke that that's for mouse doctor and not medical doctor. And so in terms of our research, we know that autism is a developmental disorder that affects the brain. So what affects the brain? We know our senses, sight, sound, smell, touch, taste, all give inputs into the brain. In fact, all parts of our bodies talk to our brains. Muscles, our heart, various different organs, including the gastrointestinal tract. There's growing literature and a growing appreciation for the gut-brain connection, and not just in autism, but in a variety of neurodevelopmental disorders. And so what is in our gut? Well, there's food, obviously, maybe some coffee, and a microbiome. The microbiome consists of 100 trillion bacteria, viruses and fungi, that inhabit our gastrointestinal tract. There are more bacterial cells in your body than there are human cells, which is quite astounding. 10,000 different species of bacteria live in our bodies with a mass of 2 to 8 or 2 to 6 pounds. The brain weighs about 3 pounds. Think about that. And so again, our research is looking in animals and asking, can we manipulate the microbiome in ways to understand how the gut-brain communicate? And so in research from our lab and from others, we know that gut bacteria affect anxiety, they affect various aspects of learning and memory, appetite, mood, and affect. But again, our lab is focused on autism. Autism, being a behavioral disorder, is diagnosed by core symptoms, as we all know, vocal and social interaction deficits, as well as repetitive behaviors. But what was quite astounding to me about a decade ago was that there are many additional comorbidities, essentially non-behavioral phenotypes, that a vast majority of these children have. One of them caught my eye, gastrointestinal symptoms. Why is it that in this classic neurological condition, anywhere between 30 and 60% of children have gastrointestinal issues? They can range from abdominal cramps, bloating, constipation, and diarrhea. This made me wonder, are there events in the gut that are part of the disease process? In the intervening years, we now have about 24 studies or so that show us that the gut microbiome is different between children with autism and neurotypical children, both in the representation of those organisms and their relative abundance. What these dozens of studies show, they're quite powerful, what they show is that there's a difference. What they don't tell us, however, is what does that difference mean? Is this change because of the disease, the change in the microbiome, a consequence of the disease, or is it a contributor to the disease? So that is the central question that we asked a few years ago, is are the changes in the microbiome actually promoting behavioral symptoms? So when you ask a bold question, you have to do a bold experiment. And so this is the experiment. First, I'll describe it to you. We took laboratory mice, standard wild-type animals, and we replaced their microbiomes with that of children with autism and neurotypical children. We transplanted gut bacteria from humans into mice. You can read the title. We can transfer all core symptoms of autism from humans into wild-type animals just by transplanting their microbiomes. And I'll take you through the data. On the far left is a test for socialization. On the y-axis is how long the mice socialize. On the x-axis is the ATEC score of the individual donor, the child with autism. ATEC, ADOS, they all apply to this graph. Are instruments, as we all know, that measure the severity of autism. Each data point is a group of animals that receive the microbiome from a single autistic child. And as you can see, the more severe the autism was in the human donor, the less the mice socialize when they receive that microbiome. It's the same with repetitive behavior. We use a marble bearing test, and we use other tests as well. As you can see, the more severely autistic, meaning the higher the ATEC score, of the donor, the more marbles these mice bury, meaning that they have more of the stereotypic behavior. Vocalization is also affected. If you compare the controls, which are microbiomes from neurotypical children transplanted to mice, and again, the more severely autistic human donors, that the mice vocalize less. They verbalize less if they received a microbiome from a severely autistic child. So I told you the brain and the gut are communicating. So how are they doing this? There are multiple conduits, multiple paths by which the gut and the brain talk to each other. The vagus nerve is one nerve that connects many parts of our body, including our gut, to our brain. And so we know that the gut sends signals to the brain via the vagus nerve. Dr. Gordon told us about immune activation. 70% of the immune cells in your body are in your gastrointestinal tract, and they're being educated by the bacteria that live there. And those immune cells traffic to the brain and they change behavior. We've shown in our laboratory that bacteria in the gut affect the levels of serotonin and other neurotransmitters that are produced in the gut, or neuropeptides like oxytocin, or even hormones like testosterone and estrogen, all involved in autism, regulated by bacteria in the gut. But what we focused on are the molecules that the gut bacteria they themselves make. We want to know what is the chemical signature of these gut bacteria. What molecules are they producing? Where are they making them? How much are they making? And how much of that affects the brain? And so going back to the mouse models now, I wanted to first tackle this problem with asking a question is, can we reverse the symptoms in mice by applying a probiotic? So again, you're looking at the vocalization test. Here I'm showing you both the number of calls that the animals produce as well as the duration of calls. And the black bars are the healthy animals, and the white bars are the quote-unquote autistic mice. And you can see that they call less, and when they do call, the duration of their calls are shorter, meaning that they're just less vocal. And the blue bars are the same animals that were treated with a probiotic that we discovered in our laboratory. And you can see that we can not only prevent the symptoms, but we can also, in graphs like this, show that we can reverse the symptoms just by applying a single human microbe to these mice. It's the same with repetitive behavior. Again, mice that have the stereotypical features of autism bury more marbles, meaning that they have more repetitive behavior. Treatment with a probiotic reduces this back down to normal levels. And what about the gut? There's several studies that show that children with autism have a syndrome called leaky gut, or at least a subset of children, which means that there's defects in the barrier integrity of their intestines, and their intestines are more permeable, potentially to molecules produced in the gut. And that's what this experiment tested. Again, in the black bars are the healthy animals, and that's at baseline, looking at their level of leakiness. And next to it, in the quote-unquote autistic animals, there's more leakiness in their intestine. And this phenotype is also corrected by treatment with a single human probiotic, meaning we can repair the leakiness in the gut by treating with a probiotic. And so now we wanted to ask about molecules. When we treated the animals with the probiotic, we asked a companion question. Can we now track the chemical signature in these animals when they're exhibiting behavioral symptoms or when they're protected from behavioral symptoms? And we found several molecules that are dysregulated between the various groups of mice. But one molecule stood out to us, a molecule called 4-ethylphenyl sulfate, or 4-EPS for short. We showed that 4-ethylphenyl sulfate is elevated in the animals that have behavioral symptoms of autism. So, at the very minimum, perhaps in a mouse, 4-EPS is a biomarker for autism. But we went one step further. We took healthy animals and we treated them with 4-EPS. We just fed them this microbial metabolite, and they developed all symptoms of autism. A single microbial molecule, again, in a mouse, can induce those symptoms. And the graph that you're looking at here is now human data. We asked, do these early studies in animals translate to people? These are samples from the CHARGE study at UC Davis, where we're able to obtain serum from 129 autistic children and 100 typically developing children. And we measured many compounds, including 4-EPS. And as you can see, there's about a quarter to a third of the autistic population that have elevated levels of this single microbial metabolite in their serum. It's a good correlation Still early days, but suggesting that what we're identifying in mice may potentially translate to people. And again, if 4-EPS is actually driving symptoms in people, maybe there's an avenue for therapeutics here. So I've summarized a lot of work in a very short amount of time, but just to give you our working model, our hypothesis of what's going on in the gut, is that there's a change in the microbiome in the autistic population. We don't know what causes that change. Again, it could be the symptoms, it could be such as gastrointestinal issues, it could be the genetics, the diet, many things can change the microbiome. But the microbiome is different, generally speaking, across the population. Coupled with that, again, in some cases, there's leakiness of the epithelium, the cells that line our gut. And when this happens, altered microbial molecules can exit the gut, get into the circulation, and in the case of 4-EPS, we now know can actually get into the brains of mice and alter their behavior. And that's what potentially may affect some of the altered behaviors in humans. One treatment, again, that we are developing through Axial Biotherapeutics is a live probiotic. Can we give this one microbial organism to children and repair their leaky gut? Which I showed you we can do in human, or we can do in the mice. And by repairing their leaky gut, we can normalize the molecules that are getting out of the gut and into the circulation. We can balance that chemical signature and potentially lead to normal brain function. Again, a lofty goal, but this is what we're working towards. So I'll leave you with a very, very provocative hypothesis, and I know I'm going out on a limb here by saying this, but you heard it here first. Right? Maybe in some cases, autism is not a disorder of the brain. Maybe it's a disorder of the gut. And if that's true, that opens up entirely new avenues for treatment. So what I've told you is that there are small molecules that are produced by the microbiome that leak out of the gut and again, get into the brain and induce behavioral symptoms in mice. And we can repair this issue by repairing the epithelium, by repairing the gut itself, and the therapy does not actually go into the brain. This may be true for other neurological conditions as well beyond autism, and these are areas that we're testing, and they may be involved in associated symptoms such as anxiety and depression. They may even be involved in neurodegenerative conditions as well. And as I've already alluded to, that if any of this is true, then potentially future drugs can be targeted to the gut, which is far easier and potentially far safer than trying to get drugs into the brain. And that's what Axial is doing, and I'm proud to be part of that. I'd like to thank the people who did the work. This is a collaboration that we started with Paul Patterson, who sadly passed away in 2014. Again, I'm a microbiologist. Paul was a neuroscientist, so he taught me a lot of things. Elaine, Gil, Brittany, Janet, many other very, very talented students and postdocs have helped. Our funding sources, Autism Speaks, the NIMH, Simons Foundation, and Axial Biotherapeutics. Thank you very much.